have some of the basic things like free energy, transfer of free energy, those kind of things. So now I will be moving ahead. So as all of you might be aware, that the process of metabolism involves of two different types, that is catabolism and anabolism. So to start with, we have this catabolism. So there are two series of reactions which are linked with the conservation of energy in chemoorganotrophs. So first one is the fermentation, the second one is the respiration. So fermentation, it is the form of anaerobic catabolism in which an organic compound is both an electron donor and an electron acceptor. And ATP is produced by substrate level phosphorylation. So in this particular case, if the catabolism is accompanied with the help of oxygen, we call it respiration. So there are organisms which are living in aerobic conditions and the another one which are living in anaerobic condition. So those microorganisms which have easy availability of oxygen, they undergo the process of respiration, and those microbes which are not having access to oxygen, they undergo fermentation. However, there is a third category of organisms also which undergo both fermentation as well as respiration. For example, yeast. So in these organisms, both the things they take place. However, it depends on the availability of oxygen. So if oxygen is available, yeast prefers to undergo respiration. However, in the absence of oxygen, fermentation is usually taking place. So now this in fermentation, ATP is produced by a mechanism called the substrate level phosphorylation. So in this process, ATP is synthesized directly from energy-rich intermediates during steps in the catabolism of the fermentable substrate. So let's see in this particular figure here. So we have compound A to B. So these are kind of intermediates where the phosphorylation takes place and it leads to the formation of intermediate rich compounds B and C. Now these intermediate rich compounds, they could lead to the conversion of ADP to ATP. However, in case of oxidative phosphorylation, ATP is produced at the expense of proton motive force. So at some point later we'll see what this proton motive force is. So for the time being, you can visualize that this is one of the bacterial organism. So in this particular organism, you can see that there is a distribution of charge. That is, the membrane is positively on the outside, inside this is the negative charge. Now this movement of charge that is facilitated by the movement of electrons. So as the electrons and the protons movement facilitates, there is a reduction of charge. This reduction of charge could generate a proton motive force. Now, this proton motive force derives the conversion of ADP to ATP. So this is what is happening in the oxidative phosphorylation. So the fermentable substrate in fermentation is both the electron donor as well as the electron acceptor. So now comes the glycolysis. So it is a common pathway for catabolism of glucose, which breaks down glucose into pyruvate. So this is also known as EMP pathway, that is the Emden-Merov-Tarnas pathway. So these are the major discoverers after whom this was named as EMP pathway. So this is usually divided into three stages. So the stage one is the preparatory reactions. So these are not redox reactions. So as I already told you, that it is the redox reactions which are taking place in the living system, which facilitates the transfer of energy or the production of energy. So this first stage is not the redox stage. There are no kind of redox reactions are going on, so they do not release energy, but they lead to the production of key intermediates of the pathway. Now the second step is the stage 2, where the redox reactions occur. So in this particular step, energy is conserved in the form of ATP. So the two molecules of pyruvate, they are formed as a result of this particular stage. However, the glycolysis is not finished at that particular step. It moves to stage third. So in the stage third also, the redox reactions occur. Once again, and it leads to the formation of fermentation products. Let's say, for example, pyruvate could be converted to other fermented products, let's say ethanol and other products. So this is a typical representation of glycolysis. So here you can see this is stage 1 where we have this glucose. So this glucose, it is converted to glucose 6 phosphate. So this rearrangement of glucose 6 phosphate leads to fructose 6 phosphate. Again, this utilization of 1 ATP, it leads to the conversion of fructose 1 6 phosphate. So now from here we have two products, that is dihydroxyacetone and glyceraldehyde 3 phosphate. So subsequently following these steps, ultimately what we get is the pyruvate. So as you can see that in this particular first step, there are two ATPs which are being utilized. However, there is no production of energy is there. In the second step, you can see that different intermediates they are produced, which ultimately leads to the formation of pyruvate. And here we have production of NADH as well as ATPs. So two ATPs and two NADH they are generated at the second step, and at the final step we again have two more ATPs. And ultimately in the stage 3, where we have two pyruvate molecules generated from the glucose, they are being converted to different fermentation products. They can be converted to ethanol, they can be converted to lactate. So if we look into the details, so during stage 1 and stage 2 of glycolysis, two ATP molecules have been consumed and 4 ATP molecules have been synthesized. So in all, 2 molecules of ATP per glucose are fermented. In stage 3, 2 NAD, they are reduced to NADH. The NADH produced in glycolysis must be oxidized back to NAD positive in order for glycolysis to continue. And this is accompanied when pyruvate is reduced to fermentation products. In yeast, pyruvate is converted to ethanol with the subsequent production of carbon dioxide, and the lactic acid production bacteria, they reduce pyruvate to lactate. So during glycolysis, glucose is consumed and 2 ATP are made, and the fermentation products they are also generated. Although these fermentation products they are of no use to the microorganism, but they are kind of interesting for the brewing industries, for the dairy. Okay, so now this respiration could be of two types: aerobic respiration and anaerobic respiration. So aerobic respiration involves oxidation using oxygen as the terminal electron acceptor, while anaerobic respiration is performed under anoxic conditions or in absence of oxygen. So electron transfer is a membrane mediated process. It has two basic functions. Number one is it facilitates the transfer of electrons from primary donor to the terminal acceptor. So those molecules which can donate electron, that is those molecules which are having high potential, they will donate electrons. Those which are having low potential participate in membrane events whose end result is energy conservation. So ultimately, it is the change in the membrane potential which drives the electromotive force that leads to the conservation of energy. So look at this particular molecule, which is flavin mononucleotide. It has two units: isohaloxosine unit and the pentose erythritol. So in these molecules, the site of oxidation and reduction is the same. So these two nitrogens are the sites for reduction as well as the oxidation. So when there is a reduction, that is two protons are accepted, we'll have NH here, and when there is oxidation, these hydrogens, these protons will be liberated and will get the nitrogen. So in some of the molecules, both oxidation and reduction will be there. So several types of oxidation reduction enzymes participate in the electron transfer. So some of the enzymes are NADH dehydrogenase, flavoproteins, iron, sulfur proteins, cytochromes, non-protein carriers such as quinones. So all these like NADH, iron, sulfur, cytochrome, they are all protein based, and these quinones, they are the non-protein based carriers. So these carriers, they are arranged in the microbial membrane in an increasingly order. The first one is the NADH, which is followed by another one, and the last one is the cytochromes. So this NADH accepts 2 electrons as well as 2 protons when oxidized to NAD+. So this 2 electron and the protons, they are then transferred to flavoprotein, which is kind of next in the chain. So ultimately, the cytochromes, which are the next or the last in this particular chain, they undergo oxidation and reduction by accepting electron and proton from this flavoproteins. There is usually a loss or gain of a single electron, which is mediated by iron. So you can see this particular reaction here, cytochrome, which is having Fe2 plus. So when it loses one of the electron, it becomes Fe3. So there is oxidation going on. So this is the kind of structure of one of the pyrroles, which is the building block of porphyrin heme structure. This is the atomic structure where the porphyrin ring is embedded inside the cytochrome C. So now these cytochromes, they can be of several types: cytochrome A, B, and C. And also there could be variation in these A, B, C also. So now it will depend on the microorganism what are the different kind of cytochromes. So look at this structure here. So this is cytochrome C, which is having the binding site, the redox site, where this porphyrin is binding, which is having iron. So now we can notice here that there are different residues like the histidine, the cysteine. So these molecules, this iron is interacting with the histidine, and this carbon molecules, they are kind of attaching with the cysteine residues. So it is the oxidation of this iron, which is from Fe2 plus to Fe3, which results in the transfer of energy. So the clusters of iron and sulfur atom with Fe2 and S2, Fe4 and S2 cluster, they are present in the cytochromes. So it is quite possible that iron is present either as divalent or as the quadruvalent iron. So one of the common one is the ferredoxin. This is a common heme iron sulfur protein. It has Fe2S2. So two molecules of iron with two molecules of sulfur. So this is a typical arrangement where two molecules of iron attach to the sulfur and they are embedded inside the cytochrome. So we can see that iron is bound to the two cysteines here and two cysteines here. And now this particular kind of arrangement would be Fe2 and S2. So we can see it's a cuboidal kind of arrangement. So in this, the cysteine linkages are again attached to the iron molecules. So the last one is the quinones. So these quinones, they are hydrophic molecules that lack a protein component. So now these compounds, they are very small, they are hydrophic and hydrophobic, and they are free to move within the membrane. So here you can see this is the particular site. Sorry. Okay, so this is the particular site which is when oxidized, you see the oxygen atoms, and when this is reduced, protons are accepted and we see hydroxyl attached here. So we can have a typical multiples of 6 to 10. So when oxidized, it requires 2 electrons and 2 protons to fully reduce. The proton motive force. So electron transport carriers, they are oriented in the membrane in such a way that electrons are transported, protons are separated from electrons. So there is movement of electrons from one enzyme to another one, and in that there is kind of separation between the electrons and the protons. So two electrons along with the two protons enter the electron transport chain from NADH. Through NADH dehydrogenase that initiates the process. So carriers in the electron transporter, they are arranged in the membrane in order of their increasing positive reduction potential, with the final carrier in the chain donating the electrons plus protons to a terminal electron acceptor such as oxygen. So during electron transport, protons are released to the outer surface of the membrane. So these protons originate from two sources. Number one is NADH, and the second one is the dissociation of water to proton and the hydroxyl in the cytoplasm. So this is a typical figure which depicts the proton motive force. So here one can see that we have this NADH which through FMN facilitate the exclusion of 4 protons. And also this is kind of facilitating the movement of electron from this whole complex to the another one complex. And similarly from complex 1 to complex 2 and to complex 4. So there is kind of movement of electrons within the complexes and there's exclusion of protons to the outside. So in a way, the membrane which was having minus 0.2 of potential, it eventually turns out to be positive because of the exclusion of the protons from the inside of the cytoplasm to the outside of the membrane. So electron transport leads to the formation of an electrochemical potential across the membrane. This potential along with the difference in pH across the membrane is called the proton motive force. And this causes the membrane to be energized. So some of the potential energy in the proton motive force is then conserved in the formation of ATP. So this variation in the potential dictates the formation of ATP. Besides ATP, this proton motive force can also be used to do work. There are different kind of works that could be done. So it could be related to the transport of biomolecules across the membrane, or it could be for the movement of the bacteria. So there could be movement of the flagella, or it could also require another kind of chemical reactions which require energy, some kind of endogenic reactions. So now we'll come to the ATP synthesis, then ultimately how the ATP are synthesized. So as I told you that this PMF creates a torque in a large protein complex that makes ATP. So this complex is called ATP synthase or ATPase in short. Now this complex consists of two units, the F1 unit which is kind of multi-protein cytoplasmic complex and that carries the chemical function, and the F0 that is responsible for the ion translocating. So here we can see that it has different kind of units. So when these protons are attached to the alpha, this AUAS unit, then this C unit has 12 subunits. So this C2 unit, it kind of rotates because a torque is generated, and this transfer the torque by this sigma and gamma subunit to the F1 unit. Now this also has like different, F1 unit has different components, like it has 3 alpha, 3 beta and delta subunits, and this torque or the rotation of this whole F1 subunit leads to the generation of ATP from ADP, or the phosphorylation of ADP leads to the formation of ATP. So as the proton enter, the dissipation of proton motive force drives the ATP synthesis. So as the protons are being moved outside of the cell membrane, it is continuously leading to the formation of the ATPase. So ATPase is reversible in that ATP hydrolysis can drive formation of a proton motive force. So if this torque is generated in this ATPase, it is leading to the formation of ATP. And however, when this sufficient amount of ATP is there, this ATP can go to the formation of ATP and leads to the opposite direction of torque. So initially, the torque was into this anti-clockwise kind of direction, and if this ATP is degraded or dephosphorated to ADP, then there will be clockwise kind of rotation of this particular ATPase. So this is how the whole organism is working in synchrony to form ATP synthesis. So probably this is where I would like to stop in this particular talk. So thank you very much for your patience.